Good morning, everyone. Today we are continuing our discussion of our non-Mendelian genetics. So we've covered things like incomplete dominance and having multiple genes, and today we are going to be talking about something called polyallelic. If polygenetic means multiple genes, then you should be able to figure out that polyallelic means multiple alleles. And as a result, you will get 3 or more phenotypes. But unlike when we had our incomplete dominance, with this one, we're not going to have a blend. So there's no middle phenotype. For example, in the game, we have our drake tails. And the way that those tails work is you have long tails are dominant to kinked tails, and kinked tails are dominant to short tails. So I'm going to write this in a new way. I'm going to say that the tail gene can have a long allele, or the tail gene could have a kinked allele, and this would result in a long tail. If I had long, that would also be long. Now, if I have a tail with a kinked allele and a tail gene with a short allele, that's going to be kinked. And the only way to get a short tail is to have TS, 2 of the short tail alleles. Now, when we're looking at this, kinked is not a middle version between long and short. It is a totally different phenotype, which means that it's not a blend, which means that I can tell, even though there are 3 different phenotypes here, I can tell that this is not an incomplete dominant situation. Because those characteristics are distinct from each other, this is a polyallelic situation. In humans, we have our HLA markers. Because if you remember, you could have like HLA A25 and you could have HLA A12. And you have 2 of those, and that's going to create your HLA markers. The next thing that we're going to be looking at is codominant. And in codominant, you also have 3 or more phenotypes. But all of the alleles are shown in the phenotype. And this one doesn't really come up in the game. So our example is going to be from chickens. There are some chickens that have feathers that are all black, and there are some that have feathers that are all white. And our black feathers we're going to label with a B, and our white feathers we're going to label with a W. And so we're going to use our code again here, where we have the gene is labeled and then the alleles are labeled separately. So I'm going to use F for feathers, and if they have 2 of the black alleles, then all feathers are black. And if they have 2 of the white allele, all feathers are white. But chickens can also have 1 of each. And if they have 1 black and 1 white allele, then they have half of their feathers are black and half are white. And this also happens in humans. And it happens with our HLA markers. Not only are there multiple alleles for our HLA markers, so you could have like 12 or 29 or 44 or 126. There are multiple alleles. All of those are numbers. They have a whole range of numbers. But because you only get 2 of them and both of them show up on your cells, your combination could be something like 1229, or your combination could be 4429. And both of those are going to show up. So your HLA markers are both polyallelic and codominant. The next thing that we're going to look at are sex-linked genes. And these are genes on the X or Y chromosome. Normally on the X chromosome. Our example of this in the game is faded and deep. And in the game, having a deep color is dominant to having a faded color. So deep is the dominant one. So let's see what happens when we have our different parents. So moms have 2 of these X chromosomes. So a mom that has X chromosome little d, X chromosome little d, is going to be faded in color. That's the way that she'll have the recessive trait. For dad to have the recessive trait, because he only has 1 X chromosome, and then he has a Y chromosome, he only needs 1 copy of that recessive allele to have the recessive faded phenotype. Moms can also be what we call a carrier. And a carrier is someone who has 1 copy of the allele for the trait, so 1 copy of the recessive version, but they don't have the recessive phenotype because they also have a copy of the dominant version. So let's see how this plays out in terms of what offspring are going to be produced. So let's see what happens when mom has that recessive trait. So when mom has the recessive trait, I'm going to need to draw my Punnett square here. And I drew it kind of wide because we've got multiple letters that have to fit in here. So dad does not have the trait. Mom does. So here, I'm going to make a girl because it's going to be X, X, and again, another girl, X, X, where this X is from dad and this X is from mom. Here are my boys. And what we can see here is that 0 of the girls have the trait, but 100% of the boys have the trait. And that's what we often see with sex-linked traits where they're on the X chromosome, is that it's going to be way more common in boys, because boys are going to be able to always get that from their mom, whereas girls, their dad can give them the dominant version, which covers it up. All of these are carriers. So let's see what happens when one of those girls has a baby. So now we're going to say mom is a carrier. So same Punnett square setup. Dad still does not have the trait. And now mom is a carrier. So she does not have it on 1 and she has the recessive allele on the other chromosome. This girl doesn't have the trait, isn't a carrier. This daughter is a carrier. This son doesn't have the trait. This son does have the trait. So let's highlight these just for ease. So here has the trait, has the trait, has the trait. So what we see now is, again, 0% of the girls have that recessive phenotype, and 50% of the boys have the recessive phenotype. But neither of our parents showed that recessive trait. So this often gets passed along. Mom doesn't even know she's a carrier, dad doesn't have it, but their sons end up with this trait because they only get one copy of that X chromosome from their mom. Let's see what happens when dad has the recessive trait. So mom doesn't have the trait, and she also is not a carrier. Dad has the recessive trait, but again, he only needs one copy of that because he has a Y chromosome instead of two X chromosomes. So here we've got carriers for daughters, but again, 0% of the girls have the trait. And 0% of the boys have the trait. So even though dad had that recessive trait, he was unable to pass on that trait to his boys because he doesn't give them an X chromosome, and he was unable to pass on that trait to his daughters in a way that showed up because mom is not a carrier. So for this part of our notes, I want to talk about blood type in humans. And I'm specifying in humans because other animals also have blood type, but it works a little bit differently depending on what kind of organism it is. So blood type in humans is pretty complicated. It's complicated because there are multiple genes involved. So we have two genes that we're going to be looking at. One of those genes is for a protein that we are going to call I, and the other gene is for something that we call an Rh factor. So our I protein has some options. You can either have IA, where you have the A allele, or you can have the B allele, or you can have the O allele. And it gets even trickier because A and B are co-dominant. And O is recessive. Then on top of that, I've got my Rh gene, which can either be positive or negative. I'm going to use some different colors to show how this works. But remember that genes code for proteins. So if I draw a blood cell, a red blood cell, hence it being red, they look kind of like this. And on the surface of this red blood cell are going to be proteins depending on what type of blood you have. Let's draw the A proteins are going to be green. So if you have the allele for A, then you are going to have some A proteins on the surface of this cell, which I am drawing as circles, and I'm making them green. So that's protein A. If I have protein B, I'm going to make those be triangles, and I'm making these in blue. So blue triangles, these are going to be my protein B. And on top of this, I can draw another one here. There are also RH factors. Those are going to be another protein. I'm going to make those, if you have the protein, then you are RH positive. So I'm going to put some little positives. So those are my RH positives. The combination of the proteins that you have on the surface of your red blood cells is going to result in this specific blood type that you have. So this person has A proteins and B proteins and RH positive proteins. So this person has a blood type of AB positive. They are AB positive because those are the proteins that they have. The way that we write the alleles for these depends on the gene. So the I alleles are going to get IA, IB, or recessive, which is little I. So somebody that has A-type blood, we're going to write the blood types here. So you could have A blood, and A blood is going to be, if you have A, because you could get two of those A alleles, or you could get one A allele and the recessive O allele, and either way, those are going to result in A-type blood. The other option is you could have B-type blood. Just like with A, you could get two copies of the B allele, or if you get one B allele and one recessive O allele, O means that you don't have the protein, so this would just have B protein, and you would have B-type blood. People that have AB blood are really rare. They have some of our A blood, some of the B, and because you only get two of these alleles, they don't have any recessive alleles because they are codominant. A and B are both made. People with O-type blood, those are going to be our recessive. They don't make any of the protein. If we add in our RH factor, so anyone that has positive blood, they are going to have either a positive allele and a positive allele, or a positive allele and a negative allele, because the negative just means that they don't make it. So in order to have negative blood, the only way to get that is to have two of the negative alleles. So let's have you test out what you know by drawing some blood cells. The first blood cell that I want you to draw is I want you to draw what O positive blood looks like. So start with a blood cell. You don't have to change color. But let's draw what O positive blood looks like. Next, I want you to draw what A negative blood looks like. So you're drawing the proteins. And then we already did AB positive. Let's draw AB negative blood. So I'm going to give you a minute to draw what those would look like, and then you can check them against mine. So O positive blood, it doesn't have any A or B proteins, but it does have RH positives. Our A-type blood, it's going to have our A proteins, but it doesn't have any of our RH proteins. It's negative for that protein. So it's just going to have those A proteins. Now our AB blood, going to have some of those A proteins, but it's also going to have B proteins, and it's negative So it's not going to have any of our RH proteins. So your drawing should look like this. Let's see if we can figure out the genotypes of these people. So my O positive blood, their genotype is going to be little I, and then they could be positive positive or positive negative. So I'm going to put a little line that means I just don't know what they are for their RH gene. My A minus, we know that they have to be one big A, but I don't know what their other one is. And I know that for their RH, they have to be minus minus. That's the only way to have that. And then our AB blood, the only way to have AB blood is to have both of those alleles. And the only way to have minus blood is to have both of the negative alleles. The reason that we talk about blood type is because it also impacts what type of donation you can get. If you are AB negative blood, you can get a bunch of different blood types donated to you. Because in the same way that your body responds to foreign HLA markers, your body also responds to foreign blood type proteins. So if you have O-type blood and you got a donation of A-type blood that has these A proteins on it, your body is going to see that A-type blood as foreign and it's going to attack it. Now, the only reason you're going to need a donation is because you need more blood. So killing off blood that you put into your body is not going to help you out. What this means is that O-type blood can only receive O-type blood. Now, if you're O positive, you could receive O positive blood or O negative blood because that positive protein only exists in the positive blood. But if you're O negative, then you can only receive O negative blood because your red blood cells don't have any of these proteins on them. If you're O negative, you don't have A proteins, B proteins, or these RH proteins. And so your body is looking for cells that don't have any of those proteins. If any of those proteins show up in your body is going to attack it as foreign. So what type of blood can somebody that is AB negative receive? Can they receive B negative blood? Sure, because their body is going to see the B and it's not going to respond. It's going to be like, yeah, I've got B proteins on my cells. That's totally fine. Can they receive AB negative blood? Yeah, that's going to work as well. Can they receive AB positive blood? No, they can't. So the A and the B proteins are fine, but if those positive proteins show up in any of their cells, then their body is going to attack it as foreign because they are not used to seeing that positive protein. As a review, blood type in humans is really complicated. Not only is it controlled by multiple genes, but those genes, at least the I gene, is polyallelic. There are 3 different alleles that you could have. And 2 of those alleles are codominant to each other, which is going to make for just really interesting combinations that show up in our blood.